NF1 is a rare genetic autosomal disease that causes tumors to grow anywhere that you have nerves along your body. The growth of tumors is highly unpredictable, and this includes when it's going to happen, where it's going to happen, and how severe the tumor is going to manifest. The Gene Therapy Initiative is one of our major research initiatives that focuses on developing a curative treatment for neurofibromatosis, which entails the correction of a mutation in the NF1 gene.
The GTI initiative is very broad. We have not only researchers that look at delivery systems to get cargo into the patient, but we have researchers producing the cargo that will go into the delivery systems.
The Gilbert Family Foundation funds high-risk, high-reward research projects. So we like to think of ourselves as the investors that come in at an early stage of research ideas and taking the roads less traveled. So we fund innovation, we fund what might seem like crazy ideas, but we don't want to leave any stone unturned for our NF1 patients.
So the Gilbert family and the GTI has been a wonderful investment in bringing together a lot of investigators to take a multitude of approaches for restoring gene function. And there are multiple ways to attack the problem. And with NF1, patients have a large number of different types of mutations. So we need different approaches. No singular genetic approach is likely to work. And so what we have been doing is the GTI has funded initially 4 different projects that I've been involved with at UAB.
We have developed new technology. We are now testing ribozymes that we know target and repair mutated NF1 in the cell. We have evolved ribozymes in human cells, so we have tried to make them better to be more active in the cell. We have tested delivery methods, so I think that overall we are on a good path.
So my lab has been working on translation termination and eukaryotes for 25 to 30 years. Along the way, we started developing small molecules that could suppress translation termination. And so what we really need is the appropriate disease model to really test our tools to see if they're going to work in a clinical setting. You know, it takes time and resources to actually test it in a genetic disease, and the GTI is providing that time and the resources.
So my lab is interested in the structure function of neurofibromin, the protein that when mutated causes neurofibromatosis type 1. And so in trying to understand that structure function, we've created cDNA systems and cell line systems to study that. And then when it comes specifically to the Gene Therapy Initiative from the Gilbert Family Foundation, my lab's been studying antisense oligos that are used to modulate splicing. One of the really nice things about the Gilbert Family Foundation is that they've essentially required us to work with experts from around the world that have expertise in other fields.
Tremendous advances have been made in diagnosis, the ability to do genome sequencing, imaging, and even treatment in recent years. But I think what the GTI has done is take a really serious sort of big picture look at neurofibromatosis. The question is, what's the root cause and what can be done to really affect what is effectively a cure for neurofibromatosis? You know, that's a level of thinking that goes way beyond what current research has tried to accomplish. And so I think, you know, their vision is a longer term one, and it is a deeper sort of approach to a condition that is, you know, for people a lifelong and completely unpredictable experience.
So the Gene Therapy Initiative, we are concerned about bringing forth a curative treatment for NF1, but also doing it in the quickest way possible, while not, of course, compromising the integrity of the development process. And one important thing that we do towards that end is taking an all shots on goal approach. So while we are early on in our journey, we are investigating a huge variety of different gene targeting strategies.
We've seen what can be done in the lab. We've seen how results can be translated from a petri dish to a subject like a mouse or a pig. And we are very confident and very excited that within the coming years that we'll be able to now do the translation from our preclinical animal models into human patients and start helping people, which is the main emphasis of what we do.
I think the future of the gene therapy initiative is quite bright. We love to share with the NF1 patient community and the caregivers and families that are affected that the state of the gene therapy initiative is good, and there's much to be hopeful about.